Okay, so it falls to me to introduce my former colleague, Andrew Young, a former colleague from Amylin Pharmaceuticals, who's now at GSK, and he's also going to speak to you about metabolic disease and peptides, where he's had a long, long history. And I don't know if he'll tell you this, but he's the scientist who saw the poster at the ADA by John Ing on exenatide, and that's what catalyzed Amylin to license in the exenatide molecule. So that was back in 1996.
Yeah.
Yeah. So very good. Andrew.
Thanks, Chris. And thanks for inviting me to this symposium. It's actually exciting that peptides are now becoming very much more significant as medicines than they were before. I neglected to put my employer's name there, GlaxoSmithKline. So although I started off life at Amylin Pharmaceuticals for the last 3.5 years, I've been working at GlaxoSmithKline. I'm fairly mindful from the show of hands before that I'm speaking mainly to a room full of chemists. So what I thought I would do is go through the attributes from a biologist's point of view that I believe contribute to commercial success of a therapeutic peptide, and specifically in the metabolic disease area. So just to define metabolic disease, we've been talking a lot about diabetes here, but it also includes many other what are really risk factors for cardiovascular disease. The largest unmet need is probably obesity. It's the toughest one to treat. Dysglycemia, Amylin's involvement was actually more with type 1 diabetes than with type 2, but then progressed to type 2 diabetes. But there are other forms of dysglycemia that are amenable to metabolic peptide therapeutics, dyslipidemias, and even hypertension. The reasons to treat metabolic diseases were initially for diabetes, both type 1 and type 2, were actually microvascular. So all of the bad things that could happen to you, and especially nephropathy, which is the most expensive complication of type 1 diabetes, about $70,000 per year per patient on dialysis. But other opathies, which were really diseases of small blood vessels. Of course, for type 2 diabetes and also for type 1, macrovascular disease is a major component of the disease burden. And here we're talking about coronary artery disease, stroke, etc. A considerable fraction of the population die from those causes. And not to be dismissed are quality of life issues. Now, the payers, the people who pay for these medicines, may not be as concerned about quality of life, but it's certainly obesity is a major detractor of quality of life. And if we can treat obesity with peptides, we will be addressing a large unmet need. So, commercial success, what does that mean? Well, it really means that you get more back from the drug than you've spent developing it. And probably, at least for obesity, that has not been the case for the industry. I mean, I haven't figured out what the return on investment must be, but there have been about 120 drugs developed for obesity. There have been about a dozen that have been approved, and by about a year ago, of those dozen, all had been removed from the market except one, which was Orlistat, and they had been removed for late-stage toxicity. So the economics of anti-obesity drug development were dismal. So if we're going to get a return on investment and have commercial success, we have to meet the needs of what are essentially multiple customers. So the payers like to see a reduction in the burden of care. The drug has to cost less than the benefit that it provides. And right now, the most expensive anti-diabetic drug on the market, which is liraglutide, costs about $14 per day of therapy. So the payers have figured out that this is sustainable, that they get $14 worth of benefit from this $14 of therapy per day, but it may not be much more than that. For obesity, the cardiovascular outcomes from obesity are actually less than they are for diabetes. So we probably can't get much more than $5 to $8 a day for an anti-obesity therapy. That, of course, depends on the efficacy, but it's a constraint that we don't hear much about, actually, the cost of goods constraint. We have to have the drug and the delivery system all coming in with a small fraction of that total return, and allow for some margin to make to recoup development costs. The prescribers, they like to see an improved ease in meeting their therapeutic targets. And insulin was not a good example. Insulin's a particularly complicated drug to administer. Has a narrow therapeutic index. It's easy to give too much or too little. And patients on insulin therapy are always between a rock and a hard place, between having too high a blood glucose and too low a blood glucose. For patients, this involves an improved quality of life and or quantity of life. And the FDA will accept a drug that improves quality of life even if it doesn't improve quantity of life. So that's worth considering. So, going on to commercial success, I think many of you have probably seen this graph or a graph like it, which really shows the disappointing performance of the pharmaceutical industry in terms of drug approvals per year. It has been decreasing over the last 3 decades, although for biologics it's actually not decreasing. And hopefully it may increase for reasons that I will go into. During the last Super Bowl, someone from Eli Lilly had put in a blog that the average cost of bringing a new drug to market was $1.3 billion, and that this would buy 371 Super Bowl ads and so on and so forth. Matthew Herper from Forbes magazine made a comment on this blog saying how ludicrous it was, because that was an underestimate of what it cost to bring a drug to market, and that it was at least $4 billion and as much as $11 billion to bring a drug to market these days. And in his comment, he used these figures, which basically looked at what the R&D spending of several large companies was, and looked at the number of drugs approved. So you can see that when you divide the R&D spending by the number of drugs approved over the last, I think that was 2 decades, that in fact the mean was something more like where's the pointer here? $5.7 billion per drug. So how might peptides help address this? The FDA only asks 2 questions. Does this drug work, and is it safe? It does not ask, will this drug be commercially successful or cost effective, or will it be reimbursable? It just says, will it work and is it safe? And efficacy and safety don't guarantee that a drug will be successful commercially. In fact, of all approved drugs, only 1 in 3 recoups the cost of development. So peptides. Although we've been talking a lot about GLP-1, my introduction to this area was with the peptide amylin, which is co-secreted with insulin from the pancreatic beta cell. And it's the basis of the name of the company, amylin. And it turned out actually to be fairly effective in working in concert with insulin. It was actually a synergen with insulin. And after, I would say, about $40 million worth of physiology experiments, we eventually found out how this peptide worked. And it was not how we thought it worked and how we thought it would work in the beginning. In fact, we went to the clinic on 4 different occasions with different hypotheses, and it was only by the skin of our teeth that we managed to eventually figure out its utility. In fact, the first action of this peptide was to be identified was gastric emptying, which it did at very low concentrations. Then it was subsequently found that it reduced food intake. It was subsequently found that it reduced glucagon secretion, and that by that it reduced production of glucose from the liver, and it was also found that it decreased digestive secretions such as acid, pancreatic enzymes, and actually decreased gallbladder contraction. So it had a spectrum of effects that it actually seemed to complement those of insulin, which in contrast is accelerating the removal of glucose from the blood. Amylin slowed the entry of glucose into the blood. This was the first realization that maybe the GI tract, or first realization for me that maybe the GI tract had a role in control of blood glucose. It turned out that, well, we were worried actually about what would happen in patients who were taking insulin if we had stopped the inflow of glucose into the blood. If they had taken an overdose of insulin, would these people be at risk for hypoglycemia? Actually, to our surprise, and this was first identified with gastric emptying, the drug stopped working during hypoglycemia. And virtually all of those actions that we saw in this previous slide, the gastric emptying, the food intake, the inhibition of digestive enzyme secretion, and the inhibition of glucagon secretion, all of those effects were overridden during hypoglycemia. That made this drug almost physician-proof, if you like. You couldn't have designed that into a drug if you'd tried. In this instance, you see the effect of amylin to suppress glucagon secretion. Here glucagon secretion is being stimulated with arginine, amino acid IV, and here you see its inhibition with amylin. But if now you induce hypoglycemia, and you'll notice that the scale here is greatly expanded, that the hypoglycemic stimulation of glucagon secretion is unaffected by the presence of this peptide. So it had some inbuilt safety. So it's got safety and it actually has efficacy. The side effect, as Wolfgang has told us, to be avoided, in fact, with almost all of these metabolic peptides, is nausea. It turns out that amylin's effects to inhibit gastric emptying, its effects to inhibit food intake, its effects to inhibit glucagon secretion, and its effects to promote nausea originate in this part of the brain, the area postrema. The area postrema is It's called a circumventricular organ. They're regarded as sensory organs of the brain. Their characteristic is that they have a low blood-brain barrier. They have fenestrated capillaries so that macromolecules up to a certain size can cross and stimulate neurons within the brain in those particular places. These particular places are not circumventricular organs, and they probably represent the receptive areas for amylin, which is also a neurotransmitter in the brain. That hypoglycemic override, it turns out that the neurons in that part of the brain that are sensitive to amylin, and here we see an example of such a neuron. This is a brain slice experiment where a 0.5 millimeter thick slice of brain has been taken out of a rat, and the neuron is firing, and when you put amylin on it, it increases its firing rate, but it also decreases its firing rate in response to the glucose that's perfusing that little bath with the brain slice in it. And it turns out that the peptide sensitivity and the glucose sensitivity are almost coincident. And this is true not just for amylin, but it's true if for example, we put GLP-1 on that brain slice. We see a similar phenomenon. So there's something more interesting about the area postrema, and that it is replete with receptors for various gut peptides. So here we see that actually these two components make an amylin receptor. Here is the brain slice cut crossways, like a coronal section now. So this is the area postrema, this little triangular-shaped part, and you can see that the same part of the brain has got receptors. This is the growth hormone secretagogue receptor, or ghrelin receptor is present there. The GLP-1 receptor is present there. PYY receptor or NPY receptor is present there, and leptin receptors, oxytocin receptor, and it transpires that all of these peptides are anorectic. So it represents an important area in the integration of nutrient flux. Now, there was some mention by Wolfgang about molecular size. It turns out that size does matter, and here is a 300 molecular weight molecule, which is fluorescein actually, and you can see that when it's perfused in an animal, that the fluorescein permeates the area postrema and ends up in the adjacent NTS, which is a adjacent part of the brain, also involved in GI function. Up to 3 kilodaltons, you can see there is good access of molecules to the area postrema. As you go above 10 kilodaltons, you see that there's greater and greater restriction of molecules to this part of the brain. It turns out that there are indeed little extravascular spaces where these larger molecules accumulate, and that some of them signal, but by the time we're getting above 70 kilodaltons, there is virtually no permeation of the macromolecule into the substance of the brain. And indeed, that may underlie the reason that these molecules have low nausea, but they also don't produce as much weight loss as smaller peptides do. So that's one thing to be aware of when designing molecules for their kinetics, is that they may not in fact, access this part of the brain, which may be part of their utility. This, of course, doesn't prevail at the beta cell. If all you want is a molecule that just is hitting the beta cell, you don't have to worry about the size because you don't have those fenestrated capillaries to worry about. So, started adding up what all of these different peptides do, and Pramlin and most of therapeutic control in the past had really been focused on accelerating the disposal of glucose from the plasma. So they were either insulins themselves, drugs that stimulated the secretion of insulin, drugs that increased the sensitivity to insulin. There were some drugs that acted to slow the appearance of nutrient, but really pramlintide, which was the amylin agonist, ended up being a drug that almost totally acted to prevent the appearance of glucose in the plasma. And that's why it worked pretty well with insulin, which worked totally to increase the disappearance of glucose from plasma. Exenatide, it turns out, did both jobs. By its insulinotropic effect, which everyone was, people were very aware of the insulinotropism of GLP-1 agonists at the time, but also, as we discovered, by slowing the appearance of glucose. So this part of it, at least, was attributable to its effects at the area postrema, and this part of it was probably attributable to its effects at the beta cell. And indeed, it became an anti-diabetic benchmark at the time that it was first identified. The best available therapy, which was like the UK PDS United, it's a large UK trial with intensive therapy of the time. This is a cumulative distribution diagram showing that people on standard therapy had a hemoglobin A1C, a chronic measure of glucose, that was in the area of about 8%. And there's a distribution around that, whereas the normal range was something less than 6.3%. Here, this was early data with 0.8 weekly injections of the long-acting release exenatide, 2 milligrams per week, and you can see it's moving this back towards the normal range, and indeed you could almost guess that 8 milligrams per week would actually normalize glucose in diabetic patients. And this, in fact, is very similar to the efficacy that's now seen with many of the GLP-1 agonists, essentially a normalization of plasma glucose. That's what made that a benchmark therapy. You saw from this that there was some benefit to be obtained from combining mechanisms of action here. And that probably is still the case. First of all, we're probably able to get efficacy that a single agent does not deliver us. We can get a potency, an in vivo potency that a single agent cannot deliver. That will reduce our cost of goods. All of the benefits. I actually regard potency as perhaps the most enabling of the attributes of a peptide molecule. Potentially, we can also amplify therapeutic index if we're getting synergism of the therapeutic effects, but we're not amplifying at the same time the adverse effects. It's worth pointing out that the main reason or a major reason to combine is because that's the way things normally happen with peptides anyway. When we have a meal, we don't secrete one peptide, we secrete them in concert. And there's reason to believe the wisdom of nature in doing that. No peptide does the same job as any other. So there indeed have been some examples, or actually many examples, of synergy between peptides published now. GSK has done some work with GLP-1 PYY synergy. Prior to that, pramlintide and leptin has a profound synergy in terms of weight loss. And amylin and CCK, this was an early study we did in rats. In fact, this is the complete response surface for a reduction in food intake. And it's hard to pick up from this, so what I've done instead is taken a transect across this surface. And we've looked at a dose of CCK that produced 70% reduction in food intake and a dose of amylin that did the same. And then just as you titrate one out and the other in, you can see that there is a profound increase in efficacy, even though we have that exceeds the additivity that might be expected. And this doesn't necessarily happen at a 1-to-1 molar ratio. In this case, it was a 6-to-1 molar ratio. But it exemplifies the potential there is there in obtaining very potent mixtures of peptides. I'm going to spend the rest of the talk talking about what we need to do to optimize peptide products. And this is kind of what I regard as the optimal plasma profile. The nausea that happens usually happens at the initiation of therapy, which unfortunately is the time when you often get a burst from a depot formulation of a peptide. So the optimal formulation would have no burst within the first 24 hours. Similarly, it would have a slow rate of change of drug concentration. If you've got wild oscillations, you may expect that to have the same detrimental effect as a burst would. And indeed, the more smooth the change in drug concentration, the less that nausea appears to be a problem. From experiments done in the clinic with exenatide, we know that if you approach maximum plasma concentration very gradually, for example, greater than 2 weeks and potentially up to 4 weeks, you can largely avert the nausea. Also, right now there are a lot of formulations that are at the weekly frequency of administration. It may be possible to have monthly dosing, and ideally this is the profile we'd want to obtain, which would mean if you had monthly dosing, you would want the second dose starting to come in as the first dose was going out, so that the sum of the two would be that profile. So are there any therapies that currently do that? Well, certainly not exenatide BID. These are the actual kinetic data for this twice daily molecule, and the recommended administration is actually to start at one dose and then double that dose after 2 or 3 weeks. These are the kinetics of that molecule, and you can see there is a large rate of change of drug concentration. Liraglutide, which is daily administration, it's got a longer half-life, is somewhat better. They recommend going up in 3 steps, but you can see that here the rate of increase is still pretty rapid in comparison to the ideal profile. Even the weekly formulation, this is albiglutide, but the Lilly drug and pegylated molecules have a very similar profile to this, are somewhat better, but you can see that they actually still have a fairly rapid increase. Dulaglutide, I believe, is the Tmax is at about 48 hours. And, this is much reduced over what I showed for exenatide, but could be improved. So the Bydureon once weekly. This is the technology, the PLGA microspheres. They're actually heterogeneous in size, but these are data from rats showing that nonetheless there is a fairly monotonic concentration dose relationship, so it was useful. A single dose produces still sizable burst, although it's less than the peak concentration. This is still problematic, and then it has this complex, a fairly complex release profile, which actually extends out to 2 months. This could be mitigated by stacking doses, and although it had maximum release rate at 2 months, if it was dosed weekly, as the Bigerion product is done, you can see that you can diminish the effect of that burst, and then you have superposition of the doses, so that you in fact have the Tmax is at about 8 weeks, and this resulted in a dramatic reduction in nausea, about a 3-fold reduction in nausea with this molecule. But there is in fact no product which can do those things which I talked about. The Bigerion achieved that at the expense of drug loading. It had to be 5% in order to minimize that burst to even what it was. It required weekly injection, even though the release profile went out for 2 months. It required 23 gauge needle and only 6.2% suspension to minimize blockage that would occur with that. And it was complex to mix up and to administer. In fact, if you look at the video online, it's 29 steps. So it's not what I would regard as an optimal profile. In addition, it took a long time to develop, and it's sort of complicated to manufacture and to scale up. What we would like is to have administration through a 28 gauge or finer needle. Essentially, that's the threshold of painlessness. It needs to be less than 1 mil, preferably 300 microliters of injection. We'd like it to be once monthly instead of once weekly. We'd like it to be pre-suspended, so only 1 step to administer instead of 29. And we'd like the drug loading to be high, and for this process to be repeatable with other peptides. As I said, there is nothing out there that does this currently. There are some potential solutions. We've heard about the Intarsia solution, which is basically a osmotic pump. It has the attribute that it has a very constant infusion rate, and they have low rates of nausea after the initiation of therapy. Of course, what you have to have is a very potent drug. You've only got 200 microliters of volume there to deliver whatever load it is you have to deliver over time. Burst minimization. I mean, one part of the burst may be attributable to those very fine microspheres that you have in the microsphere population that is made by the Alchemies technology. If you can make the microspheres very uniform in size, as is shown here, you may eliminate part of that burst. So I would say there is still considerable work to be done in delivery technologies in order to obtain what I would regard as an optimal product. So in summary, peptides, they are favorable in their efficacy, safety. Their main side effect is nausea. It can be mitigated by controlling the release profile. The delivery options are much more plentiful in molecules where you have a high in vivo potency. And what I mean by high in vivo potency is a requirement of drug that is potentially less than 100 micrograms per day. But how do you get that? The Gila monster did it for us. It created a drug that was virtually immune to peptidase degradation, and it retained at the same time its receptor potency. I'm suggesting that this is perhaps the way to obtain high in vivo potency is to have peptidase resistance while maintaining receptor potency. And at that, I'll thank you for your attention and take questions.
Thank you, Andrew, for the biology history of amylin, exenatide, pramlintide, and a little bit of insulin and peptides and metabolic disease, basically, and finally rounding us out with what is an opportunity, I think, for the future for improved product presentations in particularly the GLP-1 space, where there's a huge amount of opportunity. So I'll open the floor up for questions if there are any.
We worked a long time ago on the CCK, and then you have CCK and amylin was non-covalently bound, right?
Correct.
You mixed the two together.
Yeah.
And what was you said 61 or 81?
Well, on that diagram were 36 combinations.
Right. Yeah.
So I think if we, I don't know if we can go back to it.
It doesn't matter. So.
But at a particular strength of the mixture, the optimal ratio was kind of 6-to-1 for that particular effect.
6 what?
Yeah. Yeah, it could be different for another response.
No, 6 CCK or 6 amylin to 1 CCK.
There was 6 amylin to 1 CCK.
To 1 CCK.
Yeah, yeah.
But why you didn't pursue that for that reason? Because we killed the CCK program at Roche because and the pancreatitis in animal, we didn't know that we'll know pancreatitis. A human, we didn't have the gut to go when the animal show pancreatitis, we didn't want that. But later on turned to be okay. It was good to go to human.
Right.
But nobody pursued CCK for one reason or the other.
Right.
Maybe the combination will be good.
Yeah, I think so. Well, we know that CCK is released in you or me whenever we eat a sausage, so it doesn't cause pancreatitis at all doses. Yeah. And I think part of the potential of synergistic combinations is that you achieve much more physiologic concentrations of the ligands.
Right.
Yeah.
But they keep saying if you put 2 peptides together, you have a nightmare in the formulations and all these things.
Well, depends. I mean, let's just say you wanted to mix them in a solid form. Let's say you had microspheres. Yeah. I mean, there's no reason why there should be a problem. I grant you that there may be problems when in solution, but there are lots of alternatives to having drugs in aqueous solution.
So you recommend in solid form and then do the formulation?
Well, possibly solid form, possibly a non-aqueous diluents. I mean, of course, some people mix the agonisms in the same molecule, so you've got dual-acting agonists. You have the fibrid concept at amylin where they're linked. Yeah, I think there are lots of solutions.
Andrew is saying that we shouldn't let our CMC and product development colleagues dictate our way out of a good biological opportunity.
That's exactly what I'm saying.
They said you have to think about us before you do anything. And the CCK was CCK sulfated, right?
Yeah, there was CCK octapeptide sulfated. Yeah. I mean, there may be advantages to having some of the other biologic forms of CCK. I mean, people have only gone with the sulfated because it's probably the easiest to make.
Yes.
Yeah.
My friend, we have always the the argument, he was argument is no covalent binding, two peptide is better than the one when you can do the titration. So what is your feeling? What's better? 1-to-1 with bond like the amylin strategy with the linker, or you have let the doctor dictate 1-to-1 or 2-to-1 or 5-to-1?
I'm a sort of a believer in mimicking nature. And of course, the beta cell, when it co-secretes insulin and amylin, doesn't do it in equimolar ratios. I mean, it's typically about a 20-to-1 ratio of 20 of insulin to 1 of amylin. I don't know if that's the optimal ratio, but I wouldn't be surprised if it was. Of course, other molecules which start from a common precursor have obligatory 1-to-1 secretion. So GLP-1, GLP-2 are secreted in equimolar ratios. Yeah. I think so. And it's not necessarily the case that nature has things optimized from a pharmaceutical perspective. So I think the answer is you have to actually explore it. You have to do the experiments. The experiments may be arduous because they involve combinatorial biology, but I think that's the only way to do it.
So believing in nature, insulin is secreted with C-peptide. What's the current verdict on the importance and independent effects of C-peptide? I'm not sure about that. I know that John Warren has done a lot of work and has a company around C-peptide. But I don't know if it's actually very well conserved, C-peptide, between species. That would be a clue as to whether it had biologic importance if it was conserved evolutionarily. So I can't really say. Yeah, I don't know for sure.
The C-peptide is conserved.
Okay.
Yes.
Well, that would be an argument in favor of some biologic importance.
Well, if there are no further questions, let's thank Andrew.